We have 5 main tastes. Each taste depends on a particular receptor that's localized somewhere on the tongue. So the 5 different things that we're able to taste are bitter compounds, salty compounds, sweet compounds, sour compounds, and one more thing known as umami. And this is basically the ability to taste a particular molecule known as glutamate. So glutamate. Now these 5 tastes all depend on a particular receptor. So let's imagine that here we have a tongue, and this is the front of the tongue, this is the back of the tongue. We basically have taste buds sprinkled throughout the tongue. So these taste buds are kind of all over the place, and for the most part, they are very, very highly localized in the more anterior aspect of the tongue. So they're more localized in this anterior aspect of the tongue, more so than in the back. There are 3 different types of taste buds. There are taste buds that kind of look like this, and these are known as fungiform taste buds. I'll write that down. Fungiform taste buds. And these are the ones that are mostly found over here in the anterior part of the tongue. There are also taste buds that look like this, where the little buds are over here. And these are known as foliate taste buds. And these taste buds are mostly found over here on the side of the tongue. And finally, there are taste buds known as circumvallate taste buds, which look a little like this. And the taste buds themselves are actually over here on the sides. And these circumvallate taste buds are mostly found back here in the back of the tongue. Now, each one of these taste buds, if we actually zoom in, so if we kind of zoom in to an individual taste bud, what we would find a little pore. And this pore would have a bunch of different types of cells. It would have like one cell over here, it would have another cell right next to it, it would have third cell over here, and so on. And basically each one of these cells is responsible for one of these 5 tastes. So for example, we can take this pink cell over here to be able to detect salty things. And for example, we could imagine that this whitish cell would be able to detect sweet things, and so on and so forth. So the general idea is that each taste bud, so this thing would be a taste bud, each taste bud contains all of the different taste cells. So every single taste bud is able to detect bitter, salty, sweet, sour, and umami compounds. And these taste buds are found all around the tongue, mostly in the anterior part of the tongue. So another way to say this is that we're able to taste all 5 of these different tastes everywhere in the tongue. So what I want to talk about next is something known as a labeled lines model. So here we have a taste bud. And as I mentioned before, we have all of the different types of cells, and each one of these cells is specialized to a particular one of the 5 tastes. And what you can see down here is that each cell has a little axon projecting from it. And what is interesting is that these axons actually remain separate all the way to the brain. So all these get projected and they eventually reach the brain. And they actually synapse on different parts of the brain. So let's imagine that this is the gustatory cortex. So let's imagine that this is the part of the brain that receives input from various taste cells. Well, each one of these different axons will synapse on different parts of the brain. So here we've got this axon, it's going to synapse over here, and every other axon, every other taste cell that is this color blue, so let's just say this is a sweet cell. Let's say this is a sweet taste cell. Every single sweet taste cell will send its axon to the brain, and it will all end up in this one region of the gustatory cortex. So we can say this is the sweet region of the gustatory cortex. I keep using this word gustatory, and taste can also be thought of as our sense of gustation. So gustation. Similarly, all the cells that are green, and we can say that green could be bitter, so we can say the green cells are bitter cells, bitter taste cells, they're really bitter. They will all synapse in one part of the cortex, which we can imagine to be the bitter part of the cortex. It's very mad at the world. So this is basically how taste cells send their projections to the brain. They actually send them through dedicated axons, and there's no mixing. And this is known as the labeled lines model. So let me just write that down here, labeled lines model. And again, what the labeled lines model is saying is that each one of these cells has its own dedicated labeled line. And this distinction is basically carried on all the way to the cortex itself. Now let's look at an individual taste cell itself. So let's look at an individual taste cell. So here's the axon. This is the cell nucleus and the little hairs that kind of project out into the tongue. So what happens is, let's say that this is a sweet cell, a sweet taste cell. So it's sensitive to sweet molecules. So let's imagine that we've got a little molecule of glucose. So let's imagine you're eating cake. Some glucose will hit the tongue, and some of the glucose will find its way over to a sweet cell. And why is the cell sweet? Well, it's sweet because it actually has receptors in the membrane. So it has little receptors in the membrane that are sensitive to glucose and other sweet molecules. So when the glucose binds to the cell, it triggers a cascade of events that eventually allows the cell to depolarize and send an action potential all the way down its axon and to the brain. Now I mentioned that we have 5 different tastes. So we have sweet, umami, bitter, sour, and salty. These top 3 taste cells over here have similar receptors. And these receptors are known as G protein, so G protein coupled receptors. So basically what a G protein coupled receptor is, let me just go ahead and draw it out. It's a intermembrane protein, so this is the protein, and then let's imagine this is the cell membrane. So this protein is a receptor, and the receptor binds to a ligand. So let's imagine that we have glucose in this case, so let's imagine that a molecule of glucose comes in, hits the receptor. The receptor will undergo a conformational change that basically causes a G protein, which is coupled to it, so this is a G protein over here. So when the ligand binds to the receptor, it causes the G protein to dissociate, so this bond is broken and the G protein goes off. And the G protein can do a couple different things inside the cell. So this is inside the cell over here, this is outside the cell. So basically the G protein can go off and do a few different things. Now one of the things that the G protein can do when it gets dissociated from this receptor is it can actually open some ion channels. So the G protein can go and open ion channels. So the opening of ion channels via conformational change can actually cause the cell to depolarize and fire an action potential. So it fires an action potential. So on the other hand, these 2 taste cells, sour and salty, rely on ion channels. So instead of relying on a G protein coupled receptor, they actually rely on a receptor. So let's imagine this receptor over here. So in the case of salty, let's imagine that a molecule of NaCl, so a little salt molecule comes in, it'll bind to this ion receptor and then cause the receptor to open up. So the receptor will open up and it'll allow positive ions outside the cell to flow in. When positive ions flow inside the cell, it causes the cell again to depolarize and fire an action potential, and that action potential goes to the brain. If I, again, draw a profile of someone, here's the nose, here's the mouth, and here's the chin. There is a nostril, and this nostril basically is an opening to allow air and various odor molecules to come into the nose. And there's actually a region that you can't see, but this region is known as your olfactory epithelium. So this region is the olfactory epithelium. Olfactory epithelium. So again, olfactory, olfaction. So separating this olfactory epithelium from the brain, because the brain actually sits up here. So we've got a little bit of the brain and it sits up here. So separating the brain from the olfactory epithelium in this nasal passage over here is a piece of bone known as the cribriform plate. So this little piece of bone is known as the cribriform plate. And sitting right above the cribriform plate is actually a little extension from the brain. So there's actually an extension that comes from the brain, and it sits right above the cribriform plate. I'm drawing it in here in purple, and it looks like a little bulb, and this is actually known as the olfactory bulb. So olfactory bulb. So again, olfaction, olfactory bulb. And the olfactory bulb is basically a bundle of nerves. So it's actually one of the cranial nerves that exits the brain. And so this bundle of nerves sends little projections through the cribriform plate into the olfactory epithelium. And so this projection of nerves breaks off and branches off, and there are thousands and thousands of cells sending little connections into the olfactory epithelium. So I'm only drawing a few of them over here, but there are thousands of these cells sending little connections into the olfactory epithelium. And at the very end of each one of these little cells are receptors. And each receptor is sensitive to one particular molecule. It's actually sensitive to one particular type of molecule. So for example, let's imagine that we have a benzene ring. So we have a little benzene ring, a little odor molecule, and it travels into the nose, and it'll actually bind to a small receptor on one of these nerve endings.